Hello everyone. So far we talked about pairwise sequence alignments and we use different algorithms to deal with it. And today we are going to talk about profiles and multiple alignments. So first thing first, why do we need multiple sequence alignment? Actually, we talked about it before. To find out whether a sequence belongs to a protein family or not. What's a protein family? We talked about, for example, homologous genes and the similarities that we can find between different genes. And for proteins, we can say the same thing. There are some proteins which they have similar function, similar structures, and not necessarily 100%. It could be 80%. Sometimes they have only 10% similarity, and again we consider them inside the same family. And the way we define the similarity percentage differs from one organism to another, or even inside the same organism we have different percentages in order to compare them and consider them inside the same family. But what's the goal here? This one will lead to prediction of protein function and structure. If we can find out about such families, then of course it would be much easier for us to understand more and more about protein function and structure. And the other thing is related to discovering conserved regions. If you remember, also we talked about conserved regions, that in different organisms you can see similarities in the, for example, DNA. You can see sometimes they are 100% similar in the same position in their DNA. For example, for human and gorilla, in beta globin gene, you can see exactly these nucleotides from, for example, this position minus 153 to minus 148. They are 100% the same. But you see, even there are some differences in other organisms, but they are pretty much the same. So we are looking for such conserved regions because it most probably shows us that they have the particular function and there are similarities between them even in the function of the protein. And one way in order to organize our multiple sequences is to show them as a profile. So when we talk about the profile in bioinformatics, it means that we show our multiple sequences. It's like a graph, it's like a diagram. So assume that there are some sequences in terms of DNA, and we want to show them as a profile like this. This is the way to do that. Just look at them position by position. For example, this is the first position, this is the second one, and so on. So what are the possibilities? We have 4 nucleotides and 1 gap. And as you know, these gaps, they are the ones that we are considering ourselves. And now, in order to show them as a profile, we can show them as relative frequency. It means that, for example, for A, how many A do I see in the first position? Nothing. So 0, you can either write here 0 or just leave it empty because it doesn't have any value here. And for C, you can see 3 of them. 3 out of 5. So 60% of times you can see in these multiple sequences that C appeared. So 60%. Again, I don't see any G. And what T, I see 1 of them. So 1 over 5, it is 20%. And I see also 1 over 5 gap here. So altogether, their summation should be equal to 1. So this is a probability distribution. We will talk about it. So 60%, 20%, 20%. And here for the second position, I see all of them are A. So A for the second position, 100% of the times I can see that. So whenever you see 1, it means that for all of the sequences for this position, we have the same nucleotide. If we talked about proteins, then we had the same amino acid for each one of them. So when you represent your multiple sequences like this, we call it a profile, and later we will show you why profiles are very important. And by just looking at it, I can get idea about the multiple sequences much easier. As I said, whenever you see 1, right away you understand this position contains exactly the same nucleotide. And when you see lower ratios like this, it means that it has some percentages, but not necessarily it shows that the nucleotide should be this one. 80%, okay, this is a big value for it. So most probably it should be A, not G. And maybe this one is a mutation. All right, now it's important for us to compare a new sequence with a profile, because we want to know whether this sequence belongs to this family or not. So it could even lead to a new discovery to understand that a gene, or for example a protein, belongs to a family or not. And assume that you have such a profile, and then you are able to compare this sequence to this profile. But how do we do that? One way to deal with that is related to position-specific scoring metrics, in short PSSM. And as you can see inside this table, they show it in terms of probability distribution. Again, I will talk about probability distribution very soon. And it's not always like that. You can even show it by the frequency itself. And you can see inside this matrix, we have A, C, G, T, no gaps, because we are interested to see about only these ones. There are some tables including gaps as well, but usually that's the way we show our PSSM. And I will also show you how you can calculate the values in this slide. So let's see how we can calculate each one of the cells of our matrix. In order to find it, you should get the summation of these two factors. Let's go through all of the indices and also the notations. So I is the entity, could be nucleotide, could be amino acid, and J is the position in the profile. So position of the profile, it means that this is the first position, second one, third one, and so on. So K is the entity, but this time inside the profile, not the sequence that we want to compare it with. So I is related to the sequence, and K is related to the profile. And MIG is the element inside our PSSM that we are looking for. That's the one we want to calculate. And it is in terms of I and J. It means that the nucleotide, for example, and the position inside the profile that we are trying to find. And SIK is related to a score of aligning I. It means that, for example, nucleotide that in the sequence that we are trying to compare with the elements inside my profile. So, for example, if I'm trying to align A as a nucleotide to a nucleotide in the profile, A with C, for example, then it is very similar with the ones that we saw in the pairwise sequence alignment, and to find the score of aligning. I will show you in the example. And PKJ, it is related to relative frequency in the profile. What do we mean by relative frequencies? These values. These are probabilities, relative frequencies. Okay, so they are related to that. And let's see how we can calculate the metrics based on this profile. And I want to compare, for example, this sequence with this one. So there are some assumptions, like, we give you the score of aligning like before. Like in pairwise alignment, we also gave you these scores for dynamic programming. So for example, aligning A with C, the score is 5. A with T, this is 3. And A with a gap, this is -2. And instead of that, we can also give you the gap penalty for all of the same value. So not necessarily they are different. Sometimes we can give all the gap penalties the same value, and you saw it in different examples. Now, let's say I want to check for the first position whether if my sequence contains A for the first position, and I want to compare it with this profile, whether this is okay to have A in the first sequence or not, but doesn't matter. We just want to give it a score in the matrix, and we will analyze it later. So in this scenario, if I want to calculate this one, it means that Mij, I is A. It means that the nucleotide in my sequence, and 1 is the position that I'm looking for, I and J. Now, the summation of scoring alignment and the related relative frequency of that. So whenever you see I, you can just insert I inside it. So I have only for the scoring score alignment. So here I have A in all of my score of aligning, you see, in order not to be confused how to use it in my formula. Always try to understand what is I, what is J, what is K, and then you assign it much easier in your formula. And for K, what is K here? Entity in the profile. So one by one, I should compare it with the possible nucleotides I have in my profile. So let's take a look at this one. So for A, I don't have any value here, or it is 0. Then in that scenario, if you are multiplying 0 with something, then again it will return 0. So I don't even consider them. I consider only C, T, and the gap that I have value for. So I consider here C, T, and the gap. You see, that's the reason for K, I consider these 3. Because if you consider the rest, as I said, it will return 0, and you multiply 0 with any probability. This one again, it is 0. So aligning doesn't matter what's the score of aligning. This one is 0, the relative frequency. So it will make it 0 at the end. So I don't consider for A and also for G because I don't have them in the first position. So here, what is this one? J. J is the position in the profile. The position that I'm checking is 1. So this is how I understand about I, J, K, and we don't get confused about it. I and J is easy. I'm looking for the row A and the column 1. You see, I'm looking to find out about this value. So row A, position 1. And based on that, I can insert the indices and consider them one by one. For K, it is a little tricky because I should take a look at this one. And I understand K could be C, T, and gap. That's why we have summation, because we have other possibilities too, as you can see. So one by one, if I want to calculate them, this one is given to me. So the score of aligning is given to us, as you can see. So for this one, for example, AC, this is 5, AT is 3, and the gap penalty for A is -2. So we consider 5 here and 3 and -2. And what are these values? The relative frequencies. C1, it means that the relative frequency for the position 1 for C, this is 60%. For T, 20%. For the gap, 20%. So 60%, 20%, 20%, and that's the final result. We will practice it again in the weekly assignment. We will try to find out about all the positions. We will practice together. And this one that I showed you, that one is called linear calculation of PSSM. But we have also nonlinear version with different formula. you can take a look at it in chapter 6 of the textbook. This extra reading, I won't ask question about it in the exam, but if you are interested, you can also learn about it because it's not in the syllabus of the course. We can represent our profiles, we can show them in different formats. And one of them is showing them as logo. In that case, you can see this is the way it looks like. We can show them with different font color, and for nucleotides, so each one of the nucleotides are different from the other, so for sure each nucleotide can take one color. But when we talk about amino acids, some of them have some similarities. Maybe you show even different amino acids with the same color. For example, one group you show with blue color, the other one with yellow. That one could be helpful. And you see, for x-axis, we will have the number of positions. Position number 1, position number 2. For example, position number 4, it shows me that it contains only this nucleotide. So in my matrix, most probably it was 1. And how do I decide about the font size here? The formulas are different. There are different methods in order to find the font size. And you can see, for example, T here, the size of this is different from this one. One way is to just consider the relative frequency, and here, instead of 0, 1, 2, just consider 1 as the maximum value and consider the relative frequency to consider the font size. And just multiply it with the multiplicand, and based on that, you can construct your size. But personally, I don't prefer logos as representation of profiles because it doesn't exactly show me how much, for example, this one differs from this one. And I think pie chart could be much more useful. For example, for each position, you can have a small pie chart. And because you already show them with different colors, it's better to show them in pie chart for each one of them. But anyway, either you show them with pie chart or this way. For a small sequence, it's fine. But assume that you have such a big sequence and you want to show them as logos. That doesn't make sense, right? So for a small number of sequence, a small size of it, it's okay to show it with logos, but generally I never use them. But anyway, I wanted to show you about the logos because bioinformaticians tend to use it a lot. Okay, before talking about HMM, I want to talk about some backgrounds related to probability distribution and time series in order to understand that topic better. So what do we mean by probability distribution? For example, in this table, you can see a joint probability of X and Y. We talked about it in a statistics course in detail. Like inside this table, each cell that you look at, there are probabilities, there are relative frequencies. So each one of them are between 0 and 1. It could be also 0, it could be also 1, but it should be for sure between them, not more than that, not less than that. So for each one of the cells, I can confirm this is the case. And their summation should be always equal to 1. So these cells, as you can see, construct the probability distribution and their summation is 1. These side ones, we call them marginal probabilities. For example, for Y, you can see 40%, 30%, 15%, 15%, and their summation is 1. This row itself is a probability distribution. We call it a marginal probability distribution. Again, for this one, for X, 50%, 50%, and their summation is 1. So this is the idea of probability distribution, and this is very useful. We will also use it in our HMM model. And there's another term, time series. This one is very famous in statistics. Whenever you can define your data in terms of time, it could be useful to use time series. Not always time series can describe and analyze data related to time, but most of the time it's working properly. Like, for example, weather. For forecasting weather, time series methods are useful. Like, you know the weather for the previous day, for previous year, for previous 10 years, and based on that you can have a trend. I even know the weather in the last minute. I know the weather in yesterday. So I know the trend. The continuous trend that you can see is the time related to the weather before the current time. And then I can predict what will be the weather in the future. Of course, it will have error. It is not 100%. Even in your applications, you see, for example, it mentioned it will be rainy, and but next hour, you will check it, and no, it is sunny. So it has error, of course, but it could be accurate as much as possible, like 95% confidence, and it will be, for example, sunny in the next hour. 90% sure that tomorrow will be again sunny. In Cyprus, it's pretty easy to predict it in summer times, but these days it's a little tricky, right? Anyway, time series are working like that, and it's not just for predicting weather. We can use it for different things. For example, in two of my articles, I use it even in psychological systems. So the application of this could be very vast. We needed those two terms to talk about hidden Markov model, which is very important in our course. We will talk about it today and also next week. What is HMM? HMM is a probabilistic finite state machine with these parameters. So it contains some parameters. One of them is PI, finite number of states. So it should be finite. We don't have infinite number of PIs. These ones, we call it places also. That's why I denoted what by P. In different books, they may denote it with different letters. For initial state, usually in different systems, we consider initial state, which is important. So we can show it by P. Transition probability from one place to another place. We can call it TIJ. IJ corresponds to different places. The place I to place J. So PI to PJ, we show it by this transition. And the transitions in hidden Markov model, they are probabilities. And it's not always the same in different models related to bioinformatics, but here they consider probabilities. And the emission probability density. Probability density is related to probability distribution. So when we talk about probability density, we talk about the probability distribution. And in a specific state, so we are talking about also places, and I will show you the details in the example for sure. Now, let's talk about a very simple example, not related to bioinformatics, because hidden Markov model is not just for bioinformatics. We can use it in other courses as well. So for this simple example, let's see what it means, how I can construct this, and I will show you in details in one of the examples related to bioinformatics. But what does it mean? For example, if today is sunny, what's the probability that tomorrow will be cloudy? And I can show it by 15%. So this is a conditional probability. If I know the weather today is sunny, what's the probability that tomorrow will be sunny? Something like that. And as you can see, their summation is 1. So each one of them is a probability distribution. And how we can exactly specify which one should be a probability distribution, I will show you in one example. That one could be tricky sometimes, especially if the model is getting more complex. It could be tricky, but we will practice it together. As you can see, it can have a self loop too. It means that if today is sunny, what's the probability that tomorrow will be sunny again? 80%. You see? There's a mistype, by the way, here. It should have 1y, but it means that it is too cloudy maybe. But anyway, it's a mistype. Okay, in the next session on next Monday, I'm going to talk about algorithms related to multiple sequence, and I will show you also one example related to OHMM, how we can construct them, because this one could be very useful for us, and for sure we should practice it. And I will give you even one weekly assignment related to that 2 weeks later. We want to practice it because it will be one of the topics in your midterm exam as well, so we should practice it. The idea is that we have multiple sequences and we want to construct such a OHMM. In order to do that, first we should transform everything into match states, and then we can construct the whole thing by considering insert and delete states. They are related to the mutations that I told you about having extra nucleotides in your DNA, or by maybe evolution they got deleted. So we can include all of them here, and how I can understand about them is related to the gaps that you can see here, sometimes extra nucleotides. So if you just listen to it carefully next week, I'm sure it will be very easy, and this topic would be very interesting for you. This is very useful. This is not the only model. I will talk about different algorithms next week. But for example, personally, I don't use them. I use Petri nets. I will talk about it next week too, briefly, because it's not in the syllabus of your course. But for sure, I will talk about OHMM in detail and how we can construct. Okay, guys, if you have any questions, please let me know. Otherwise, I will see you guys on Wednesday and we will deal with the weekly assignment.